As I told you last time, that the modality of nervous control is not complete yet. So, dear, let's not forget about the gastrointestinal sensory nervous system. You should know that many efferent sensory nerve fibers innervate the gut, and cell bodies of these efferent fibers are located in enteric nervous system or dorsal root ganglia of spinal cord. Keep in mind that sensory nerve endings originate in the gastrointestinal epithelium or the gut wall, and from here the efferent signals, they are sent to both the plexuses of enteric system. And in addition to enteric system, the afferents, they are also sent to prevertebral ganglia of the sympathetic system, spinal cord, and also to brain stem in vagus nerves. As a matter of fact, 80% of the fibers in vagus nerve, they are efferent. Now, the stimulation of gut receptors alters the activity of gastrointestinal effector cells by either eliciting neural reflexes or the secretion of hormones. And that's how the sensations, they are exerted through enteric nervous system. As far as the receptors of the GI tract are concerned, these mainly are mechanoreceptors and chemoreceptors. And these receptors, they respond to almost anything, to stretch, osmolarity, and pH. Also, the presence of substrate and the end products of digestion. So, as a result, they initiate the reflexes that either activate or inhibit the digestive glands. Plus, these reflexes, they mix lumen contents and move them along. But first, you must know that which fibers are involved. So the sensory efferents are the splanchnic nerves, which are the mixed nerves, and the vagal nerves. As I already told you that 80% fibers of vagus nerve, they are efferent. Now the sensory fibers of these nerves, they can be stimulated by the irritation of gut mucosa or excessive distension of the gut or the presence of specific chemical substances in gut. And then the signals through these fibers, they can cause the excitation or the inhibition of intestinal movements or excretion. So, what are the functions of this gastrointestinal sensory system? You see, they can elicit local reflexes within the gut wall itself, or even they can cause reflexes that are related to gut from either the prevertebral ganglia or the basal regions of brain. Uh, Here the mention of reflex, it leads us to a very important topic of gastrointestinal physiology, which is reflex control of gastrointestinal tract. So let's start with that. There are three types of reflexes which are essential to the gastrointestinal control. And these reflexes are supported not only by the anatomical arrangement of the enteric nervous system, but they are also supported by the connection of this enteric nervous system with the sympathetic and the parasympathetic system. Generally, we classify the gastrointestinal reflexes into three types. The ones which are solely gut wall reflexes, then a reflex from gut to ganglia, and then back to the gut. And finally, the reflex type, which starts from gut, goes to central nervous system, and then back to gut. And now you need to know a little detail of each type of reflex so that you could write something in your exam, because this topic, it may be a university question. The reflexes that occur entirely within the enteric nervous system of gastrointestinal tract, they control the gastrointestinal secretions, peristalsis, and mixing. And they also control the local inhibitory effects. Now, the ones from gastrointestinal tract to prevertebral sympathetic ganglia and then back to the gastrointestinal tract, they transmit the signals long distance to other areas of gastrointestinal tract. Here I give you a few examples, such as gastrocolic reflex, through which the signals from the stomach, they cause the evacuation of colon. Then there is enterogastric reflex, in which the signals from the colon and small intestine, they are used to inhibit stomach motility and secretion. The colono-ileal reflex is another example through which the signals from colon, they inhibit the emptying of ileal contents into the colon. Now the final type of reflexes are from the gastrointestinal tract to spinal cord or the brainstem, and then back to the gastrointestinal tract. And the examples, they include the reflexes from stomach and duodenum to the brainstem, and then back to stomach by way of vagus nerves. Pain reflex is another example which causes general inhibition of entire gastrointestinal tract. But the most important example is the defecation reflex, which travels from colon and rectum to the spinal cord, and the signals from spinal cord produce powerful colonic, rectal, and abdominal contractions which are required for defecation. So you can see that how intricately our enteric nervous system is connected to the higher centers. But the enteric circuits, they could be independent also. Parasympathetic nervous system and sympathetic nervous system nerves, they usually synapse with the components of enteric nervous system. And enteric nervous system nerves, they are organized into myenteric and submucosal plexuses. But many gastrointestinal actions, they are regulated solely by the neural circuits in which a mechanoreceptor or a chemoreceptor that is stimulated in mucosa and transfer these signals back to the neurons in submucosal plexus, which stimulate other neurons in the submucosal and myenteric plexus, and finally that results into the regulation of endocrine or secretory cells. Now the interesting bit is that the neurons of the enteric nervous system, they are supported by enteric glial cells, which are structurally and functionally resemble the astrocytes in brain. In this figure, you can see two types of enteric neurons. Uh, so they are located either in the myenteric plexus or submucosal plexus preferentially. And one more interesting thing is the application of certain toxins such as tetrodotoxin. It will abolish the action potential in S type of enteric neurons. Now, here is a long list of different types of neurotransmitters secreted by enteric neurons, and some of which I have already mentioned in the previous lecture. You can see that some of them are excitatory, others inhibitory, and yet some other which have multiple functions. And then there are some which have specific functions. Say a few interesting ones are substance P, which contracts the wall muscle and increases the salivary secretions. Then there are enkephalins, which constrict the circular muscle around the sphincters and decrease the intestinal secretions. Another one is gastric releasing peptide. Commonly it is called GRP, and it acts on the glands only and increases the gastrin secretion. There is also neuropeptide Y, which relaxes the wall muscle and decreases the intestinal secretions. Now these neurotransmitters which are involved in the gastrointestinal regulation, they can be classified under different criteria also, as you can see in this list. But more interesting thing is that how a variety of action is achieved through them. You can use the example of submucosal plexus, where secretory neurons primarily use VIP and acetylcholine as neurotransmitters, and sensory nerves, they use substance P. Also take the example of myenteric plexus, where the motor neurons, they use acetylcholine and nitric oxide. Sensory neurons use substance P, and the interneurons, they use acetylcholine and serotonin. Still, the pharmacological importance of certain enteric neurotransmitters is extensive because they are used elsewhere in the body also. Now, take an example of a person on the serotonin reuptake inhibitors. And what are these? In common language, these are antidepressants. Now, these drugs, they alter the serotonin levels, regulating the gastrointestinal tract also. So the patient may experience decreased gastrointestinal motility as a side effect. All right. Now, let me introduce you to the neuromuscular junctions in gastrointestinal tract. It is the site where neurotransmitters are released from axons of motor neurons to act on smooth muscle fibers, on the interstitial cells of Cajal, and also glands and blood vessels. Histologically, these are simpler structures than the motor end plates of skeletal muscles, and the neurotransmitters, they are released from multiple varicosities of the motor axon, which they spread out along the axon. Uh, you can consider it as an adaptation of simultaneous application of the neurotransmitter to a large number of muscle fibers from a small number of motor axons. Now, the synaptic transmission itself in the enteric nervous system, it is modulated through presynaptic and postsynaptic actions. So first, we move on to the postsynaptic actions, where the slow excitatory postsynaptic potentials, they cause long-lasting responses of gut effectors during physiological stimuli. And the fast excitatory potentials, they cause rapid transfer of information between the elements of enteric microcircuits. Well, the slow inhibitory postsynaptic potentials, they also exist. For example, the shunting of blood by the sympathetic stimulation during exercise. Now, what about the presynaptic potentials? You see, the presynaptic inhibition, it's a mechanism for selective shutdown of a microcircuit. While the presynaptic facilitation, it is selective enhancement. With that, my dear students, it was all about the nervous regulation of the gastrointestinal functions. And I want to shift to the description of non-neural regulation now, which could be endocrine regulation, where the endocrine, enteroendocrine cells, they secrete regulatory peptide or hormones that travel via bloodstream to the remote target organs. For example, gastrin or secretin. Or it could be paracrine regulation, in which the regulatory peptide secreted by the enteroendocrine cell, it acts on a nearby target cell by diffusion through the interstitial space. And the examples, they include histamine, and then there is a 5-hydroxytryptamine. So, uh, let's start with the hormonal control first. As I already told you, that the gut hormones, they constitute a group of hormones which are secreted by the enteroendocrine cells in the stomach, pancreas, and small intestine that control the various functions of digestive organs. Although these enteroendocrine cells, they do not form the endocrine glands, but they are spread throughout the digestive tract, and they exert their autocrine and the paracrine actions that integrate all of the gastrointestinal function. Now, the gastrointestinal hormones, they are released into portal circulation, and they exert the physiological actions on target cells with the specific receptors for the hormone. And these effects of the hormones, they persist even after all the nervous connection they have been severed. Here is the family tree, you can see. A family tree of the gastrointestinal hormones, but we will discuss only the important ones. So let's start with the gastrin. It is a polypeptide hormone in nature, and it is secreted in two forms, G34 and G17. It is produced by G cells in the antral portion of the gastric mucosa during the gastric phase of gastric excretion. But it is also secreted in the duodenum and jejunum during the intestinal phase. The secretion of gastrin is stimulated by the presence of food in stomach, especially the peptides and the amino acids, or the stimulation of local nervous plexus in stomach and intestine, and also stimulated by the vasovagal reflex during the gastric phase of gastric excretion. Now, for gastrin, there are some inhibitory factors also, and the luminal ones of these inhibitory factors, they are the acid and somatostatin, while in the blood these are secretin, GIP, VIP, glucagon, and calcitonin. Now see that the release is inhibited by highly acidic pH, like less than 2. And this acid in the antrum, it inhibits gastric secretion by two ways. Either by the direct action on the G cells or by stimulating the release of somatostatin by D cell. So you can guess that in condition where the parietal cells, they are damaged, like in pernicious anemia, the gastrin levels, they would be elevated. Now jot down the actions of gastrin, because these are important even for the viva questions and the other stuff for the MCQs. So, gastrin stimulates gastric glands to secrete gastric juice with more pepsin and hydrochloric acid, because it induces the insertion of potassium hydrogen ATPase pumps into the apical membrane of parietal cells. And these cells, they in turn increase the hydrogen ion release. Then the gastrin accelerates the gastric motility, and may also promote the growth of gastric mucosa. More important is that it stimulates the pancreatic juice secretion. Zollinger-Ellison syndrome or gastrinoma, that's a condition in which the gastrin levels, they are abnormally elevated. Now in Zollinger-Ellison syndrome, gastrin is produced at excessive levels, often by the gastrinoma of the duodenum or the pancreas. It usually occurs in autoimmune gastritis, where the immune system attacks the parietal cells, resulting in hypochlorhydria, which causes elevated gastrin levels in attempt to compensate. But eventually, all the parietal cells, they are lost, and hence the loss of negative feedback on the gastrin also. Now there is Pentagestrine also, which is synthetic gastrin. It is composed of terminal 4 amino acids of natural gastrin plus the amino acid alanine. And it has all the physiological properties of natural gastrin. Now, next very important hormone, that is cholecystokinin, which is again peptide in nature, and it contains around 30, not around, exactly 33 amino acids. It is secreted by eye cells of the mucosa of upper small intestine, especially the duodenum and jejunum. But a small quantity is also secreted in ileum. And the secretion is stimulated by the products of protein and fat digestion and also acid. Coming to the actions of cholecystokinin, um, it induces pancreatic juice secretions, which is rich in pancreatic enzyme and bicarbonate. CCK also causes the contraction of gallbladder and relaxation of the sphincter of Oddi. Then it inhibits the gastric emptying and gastric motility. And it also augments the contraction of pyloric sphincter, while it increases the motility of the intestine. There was another effect. Mm, this CCK, this also has a tropic effect on the pancreas. So as a whole, it is responsible for stimulating the digestion of fat and protein. And it also induces satiety by acting through hypothalamus. Now, there must be some factors which govern the release of cholecystokinin. The most potent stimulator is lipid, while peptones and amino acids, they also increase its secretion. And then it is secreted in response to the cholecystokinin releasing factor also. You should know that once the cholecystokinin is released, it brings a positive feedback where, because of the enzyme release, more digestive products are formed, which bring about even more cholecystokinin, and it stops only when these digestive products move to the next part. The next one is secretin. Again, it's a peptide hormone in nature, and its structure is very similar to glucagon, VIP, GIP, and it is stored in an inactive form which is called prosecretin. Its half-life is around 5 minutes. Now, secretin is secreted by S cells which are located in the mucosa of upper small intestine, all parts, duodenum, jejunum, and ileum. Now, the secretion is increased by products of protein digestion and bile acid, fatty food, and increased acidity in the duodenal contents, like when the pH is less than 5 or even less than 4.5. And secretin's release is inhibited by again by the somatostatin. Now the actions. Okay, I would suggest you to make a chart or flashcards to memorize these actions of all the GI hormones. Well, that's up to you. So the secretin, it stimulates pepsin, pancreatic bicarbonate, and biliary bicarbonate secretion. And it also has trophic action on the exocrine pancreas. Then it inhibits not only the gastric acid secretion but also its motility and emptying by causing constriction of the pyloric sphincter. You may find it interesting that it enhances the actions of cholecystokinin on pancreatic secretion. Now, this hormone may sound new to you. Motilin is a polypeptide in nature containing 22 amino acids, and it is secreted by enterochromaffin cells and M cells in the mucosa of stomach, duodenum, and jejunum. The interesting thing is that its levels are increased in interdigestive periods, and when food is ingested, there is a suppression of the secretion of motilin. In fact, motilin is a regulator of the migrating motor complexes, or you can call it MMCs, that I will explain later. Anyway, so like that, it controls the motility of gastrointestinal tract between the meals. The other actions include accelerating the gastric emptying. Then it increases mixing and propulsive movements of small intestine and increases peristalsis in colon. Keeping an account of these actions, physicians sometimes they prescribe erythromycin to treat intestinal hypomotility because this drug, with its motilin receptors activating property, it may help in diabetic gastroparesis and dyspepsia. Moving on to somatostatin, which basically is a growth hormone, inhibitory hormone. It is again peptide in nature, and it was first found in hypothalamus. But in gastrointestinal tract, it is secreted by D cells, precisely the precisely stomach and upper part of small intestine. So you can see that it is presented in two forms. The somatostatin 14 from the hypothalamus and somatostatin 28 from the gastrointestinal tract. Its secretion is stimulated by the acid in the lumen and also the presence of chyme with glucose and proteins in stomach and small intestine. And what actions it exerts? Well, one word, uh, inhibition, okay? Because it is a statin. It inhibits the secretion of gastrin, secretin, GIT, and motilin. Also inhibits the secretion of exocrine pancreas. Actions of somatostatin on the gastrointestinal tract, they result in increased fluid absorption and decreased secretion from intestine, as well as decreased bile flow and gallbladder contraction. Then there is decreased gastric acid secretion and motility, plus decreased absorption of glucose, amino acids, and triglycerides. Now, mentioning inhibition brings me to another hormone, GIP, which stands for gastric inhibitory peptide. Well, its new name is glucose dependent insulinotropic polypeptide, and it's a member of the secretin family. Now, it is secreted by the K cells in the mucosa of duodenum and jejunum. And GIP receptors, they are found on the beta cells of pancreas. And the secretion is stimulated by glucose and fats. Now, what's your guess about the actions of this hormone? Although the name already is very suggestive. Yes, it stimulates insulin release and inhibits gastric acid secretion by directly inhibiting the parietal cells or indirectly inhibit the gastrin release via somatostatin. And then it has a mild effect in decreasing the gastric motility also. Well, we got a VIP now. This vasoactive intestinal peptide, it contains 28 amino acids with a half-life of 2 minutes in circulation. It is released in response to esophageal and gastric distension, also to vagal stimulation, and to the fatty acid and ethanol in duodenum. Keep in mind that amino acid and glucose, they do not affect the VIP release. Now, I will just narrate the actions of VIP, and if you want, you can take notes. Uh, this hormone seems to induce smooth muscle relaxation in stomach and gallbladder, and also stimulate the secretion of water in the pancreatic juice and bile. Then it causes the inhibition of gastric acid secretion and absorption from the intestinal lumen. It also stimulates pepsinogen secretion from the chief cells. Interestingly, VIP, it is also found in heart, and it causes a coronary vasodilation through its effect on the cardiovascular system. So, here comes the famous Ghrelin. It's a recently discovered peptide hormone, and it is synthesized by the epithelial cells in the fundus of stomach. Now, mainly it is secreted by the oxyntic cells in the mucosa of stomach. Its secretion is increased during fasting, and the secretion is decreased when the stomach is full. Now, write down the actions. Number one, it promotes the secretion of growth hormone. Then, it induces appetite and food intake by acting via feeding center in the hypothalamus. And then it also stimulates the gastric emptying. And now we are left with a minor one, which is peptide Yy. And for this hormone, fat is the major stimulant. It is released from the jejunum. And what does it do? It inhibits the gastrointestinal motility. And finally, here is a list of some other hormones which are believed to act on the gastrointestinal tract because they are secreted by the mucosa of our GIT. Uh, Now, if I assume right, very few of you might have drawn a chart of actions. So, as the rest of you expected, here I made it for you, but I have included only major gastrointestinal hormones. And this could help you in memorizing the actions. Other than hormones, gastrointestinal tract, it is affected by non-neural signaling molecules, and these molecules, they act in a paracrine fashion. The examples of these molecules, they may include histamine, prostaglandins, and also somatostatin. Hmm. And once again, I, and only I, make the effort to make a chart for you. Now, in this chart, you can see that histamine is released in stomach, whereas both the prostaglandins and the somatostatin, they are more widespread in their release and also in their actions. Well, now students, it is my turn to ask you a question. It might have come to your observation that when someone takes an aspirin, it usually is followed by heart burning sensation. So why is that so? This could be a viva question. If I am the examiner, I would definitely ask that. So give me the answer. Well, you see, the prostaglandins, they are cyclooxygenase products derived from arachidonic acid. Now, as a whole, it is the muscular contractions which mix and move forward the contents of digestive tract. But in fasting state, motility is achieved by migrating motor complexes, also called MMC. However, we will discuss this MMC later. Now, in the fed state, two types of movements occur in digestive tract. First type is the propulsive movement in the form of wave-like contractions, which commonly known as peristalsis. And the other type is mixing movements, also called segmentations. To elaborate, here is a chart of differences between these two types of movements. Here you can see that propulsive movement is the basic movement of GIT that pushes the contents forward through the digestive tract. It occurs at an appropriate rate to accommodate digestion and absorption of food and gut. Keep in mind that the propulsive movements are modified in different parts of GIT for proper functioning, as you can see in this chart. But the basic propulsive movement of GIT is peristalsis. Now, how do we define peristalsis? It is the involuntary constriction and relaxation of muscles of intestine, creating wave-like movements which push the contents forward. So, what could stimulate this peristalsis? The stimuli range from distension of the gut to the physicochemical irritation of gut epithelium. Here include a strong parasympathetic signal also to this list. As the usual stimulus for peristalsis is distension of gut, which results in stretching of gut wall, there is stimulation of enteric nervous system due to this stretch. So, a contractile ring appears in circular muscle 2 to 3 centimeter behind the point of distension around the gut and then moves forward. While the segment of the gut distal to the contractile ring relaxes. As a result, any material in front of the contractile ring moves forward. Since peristalsis is mediated by myenteric plexus, so it does not occur in the segment of GIT that has congenital absence of myenteric plexus. Here you should recall this pathology I told you in previous lectures. Then also peristalsis is greatly depressed or even completely blocked by anticholinergics like atropine. Interestingly, there are other body tubes also which show peristaltic activity, and those include bile ducts, glandular ducts, and ureters. Is peristalsis a reflex? Oh, yes, and it is called myenteric reflex also. It's a complex pattern of appearance of contractile ring on or at site of distended segment along with receptive relaxation. Now, what is this receptive relaxation? It is the relaxation of several centimeters of gut wall ahead of distended part, means in the area towards anus. And this relaxation allows the food to be propelled more easily. This peristaltic reflex and the anal direction of the movement of peristalsis is called law of gut. Here keep in mind that peristalsis can occur in either direction from stimulated point, but it normally dies out rapidly in orate direction while continuing for a considerable distance of 5 to 10 centimeters towards anus. Now say hello to the mixing movements. These movements, they are local intermittent constrictive contractions occurring every few centimeters in the gut wall. These movements differ in different parts of the alimentary tract and they usually last for only 5 to 30 seconds. One more thing, that the areas of the appearance of these mixing constrictions, they keep shuffling from different points of gut. And for proper mixing, these movements are modified in different parts of GIT. Although the name is suggestive, but can you list the functions of mixing movements? Well, I will do it for you. They chop and shear the contents of GIT, they mix the food with the digestive juices, and they facilitate the absorption by exposing all parts of gastrointestinal contents to absorptive surfaces. An interesting bit in this is that in some areas, the peristaltic contractions cause most of the mixing. And it is especially true when the forward progression of intestinal contents is blocked by a sphincter. So in this case, the peristaltic wave, it can only churn the contents instead of moving them forward. Now, let's start with chapter 64. That will show you the propulsion and mixing of food along the alimentary tract, starting with the food ingestion. The ingestion of food is principally determined by hunger and appetite. Why these two mechanisms are extremely important? Because they not only maintain an adequate nutritional supply for body but also determine the amount and type of food ingested. I hope you know the difference between hunger and appetite. Hunger is the intrinsic desire for food, while appetite is the preference for a particular type of food. Now, hunger, it's savage. Well, it is associated with certain effects like craving for food, then rhythmical contractions of stomach, and even restlessness, which causes the individual to seek food. For the control of food intake, our hypothalamus contains a hunger center. But there are several neuronal centers of hypothalamus which participate in the control of food intake. And these centers are like lateral nuclei, paraventricular nuclei, dorsomedial nuclei, and arcuate nuclei. Although feeding behavior is influenced by some other factors which include neural signals from GIT about stomach filling and chemical signals from nutrients in blood. Also, the signals of sight, smell, and taste from the cerebral cortex, and then the signals from the GI hormones and the hormones which are released by adipose tissue. Our hypothalamic centers, they have high density of receptors for the neurotransmitters and hormones. And hunger center operates by exciting the motor drives to search for food. Now the lateral nuclei of hypothalamus, they serve as feeding center. So stimulation of this area causes the person to eat voraciously, and that condition is named hyperphagia. For same reason, destruction of lateral hypothalamus causes lack of desire for food. What about the other nuclei? You see, the lesions of paraventricular nuclei often cause excessive eating, while that of the dorsomedial nuclei usually depress eating behavior. Arcuate nuclei receive signals by certain hormones of GIT and adipose tissue to regulate the food intake. Here, I wouldn't skip the mention of satiety. It is the state of being fed to or beyond capacity. A satiety center is located in the ventromedial nucleus of hypothalamus, and it inhibits the appetite even in the presence of food. Also, the arcuate nucleus has various neurons which project onto the satiety center.